Done. Okay, very good afternoon to everyone, and welcome to the vertical 2 of Drug Discovery Hackathon training program. And today we have with us Dr. Raman Parkesh. Raman Parkesh currently holds principal scientist position in Institute of Microbial Technology, a premier CSIR institute. He obtained his BSc and MSc from University of Jammu. He completed PhD from Trinity College Dublin and went to Oxford University and the Scripps Research Institute for postdoctoral works. His current research interest includes computational medicinal chemistry, chemoinformatics, epigenetics, RNA drug discovery, supramolecular chemistry, fluorescent chemosensors for biologically important ions, solid phase peptides and peptide synthesis, and the development of small molecule probes for various infections and neurodegenerative diseases. Dr. Raman is the inventor on the patent for design and synthesis of nicotinic acid adenine dinucleotide phosphate NAADP antagonist, which is now commercially success. Raman has published high impact journals such as Nature Chemical Biology, JACS, Nucleic Acid Research, etc. Dr. Raman has won various fellowships and awards such as big awards from Direct DBT, Oxford University Merit Award, CSIR Quick Hire Fellowship, Trinity College Postgraduate Award, National Science Talent Promotion, and CSIR UGC Net Fellowship. In today's vertical, he will be talking about targeting RNA with small molecules. So I now hand over this session to Dr. Ramesh Parkesh.
Okay, thanks there for this introduction, and thanks Professor Shastri for giving me the chance to speak on this topic. I will just want to show today that RNA is also something which we forget about targeting. So we always think about targeting proteins, and if you look at currently, 100% of the drugs are for the proteins, but it's changing now, and RNA is becoming an important target to look for. So just before I go into this, how we can target RNA region for bacterial membrane disruption, we also look for random libraries, how we can use them, polyamines, small molecules, and RNA, of course, is one of the areas we are working on. So today I will focus on how we can actually design some specific molecule for targeting RNA, particularly small molecule. And then I will briefly touch upon what additional things we can do with the RNA. So it's an interesting target. That's what my aim will be, to actually give you a little bit of an overview about this RNA as a drug target, what we have done, how the field is progressing. So I actually begin with this slide. In fact, I asked a reverse question. So why not RNA? So somebody will say why RNA, but I just will go reversely to say why not RNA. So when you want to target something, so you look at its importance, and in fact, RNA is a pretty important molecule. If you compare RNA with the protein, so only 3% of the RNA codes for the proteins. It means almost 90% of the RNA is non-coding. So it's a huge chunk of the molecules which we can target. And those proteins, out of, if you look, say 3% of the RNA goes into the protein, out of that, only a small percentage of the proteins are actually druggable. So you have a lot of the proteins which can't find any pocket or any cavity where you can put in a small molecule. So that makes actually more interesting. So if you, rather than targeting the protein, if you go back and target RNA, that will be a much better choice. So that you don't have that functional protein and you are able to mitigate the disease. And I will actually show in some of the slides that how we have used small molecules to target RNA for different metabolic diseases, triplet repeat disorders, and give you some example. But before we go into actual, logistics of how we are going to target RNA, I just want to show this slide that RNA is actually not a very, you can say it's a very rigid molecule. It's like a noodle. You can have flip-flops. It can be a single-stranded, double-stranded. You can have nucleotide bulges, loops, internal loops. And if you look at one of the slides which I show here, group 1 intron tetrahymenia. So you look how complex it. So many loops, so many junctions. So a lot of say cavities are available for targeting. And if you look 3D structure, so that gives you a overview. So how many good bulges as well as cavities are there we can target. So in terms of structural diversity, RNA is as good as proteins. So you can pretty much use that information to target with a small molecule. And in fact, it has been done. For example, if you look at the antibiotics you have been taking, so most of them actually act on the ribosome. For example, this linezolid, it's a classical poster molecule which everyone puts up to show that yes, it is possible to target selectively the RNA. Though we can say it's a ribosome, and ribosome behaves most like a protein, but then it is an RNA. So you can actually, and there is also a recent paper in 2017 where cryo-EM structure of the linezolid derivatives have been solved in targeting 50S RNA. So it is possible to target selectively the RNA, and I give you another example where they have targeted ribose switches. So Dean Shah group and Baker group in US, they are heavily using some of the computational techniques to target ribose switches. So it is possible, and also just a few years back, there was a very interesting paper where they were able to actually block or they were able to rescue the function of SMN2 gene so that they have a functional protein if SMN1 is not able to, for some reason, able to produce the actual protein. So you can actually modulate SMN2 and you can get a functional protein, and that actually was able to rescue some of the SMA mice. And this drug they use is brenaplam. This is now in the phase 2, so it is especially targeting the RNA molecules. So it goes and binds there, as you can see from this slide. So this was one interesting paper. Then there was an additional paper from the Merck where they have actually used a small molecule to target bacterial mRNA, which are like ribose switches. So they are important for riboflavin synthesis in the bacteria. So if you can block them, you are able to actually prevent the bacterial growth. So this was an important paper from Merck. This also is undergoing clinical trial. So in these two papers in 2015 have clearly shown that yes, it is possible to target say small RNAs. So that's what my talk will be on that. And in fact, this is the data from same group, ribosyl, where they have clearly shown that they are able to target the aptamer domain of the ribose switch, and that prevents the expression of the gene. So such molecules are there, and it is possible to selectively target. So this is the good news. And in fact, we also have done, we have published in JAX as well as biochemistry and ACS chem biology that it is possible to target those RNA transcripts which are involved, for example, in myotonic dystrophy as well as Huntington disease. So we have used a different combination of techniques, but we have shown, yes, it is indeed possible to selectively target that. So before we go about targeting RNA, in fact, you have to ask yourself, like, what you are targeting and what you want to achieve. So first, you want to know what are the RNA targets. So you have to know about the disease conditions. So what are the challenges? So as I told you before, that RNA is not your typical protein molecule where you can go and find cavities. It's actually a noodle-like molecule. But yes, and you know that when you want to eat a noodle, it's very difficult. But if you have the right chopsticks, you can enjoy your noodles nicely. So it means if you have a right kind of molecules, you have right kind of tools, yes, you can target RNA with the small molecules. And then there are additional things. Once you have the molecule, how you can actually go about optimizing the efficacy of the molecules and have a pretty active in vitro as well as in vivo molecules. So these are the questions which you need to consider before you go about targeting any RNA. And it's an interesting area. It's expanding, and if we can target, say, any messenger RNA, it will be great. Then it means you can pretty much control every protein. You don't need to go to the protein level. You can just stop it at the RNA level. So that's what so the aim is there. So I will actually show you what we have done, and here is this small baby Joshua suffering from myotonic dystrophy, which is a terrible disease, and it's a genetic disease. So in terms of the DNA, there is a CTG repeats, and in terms of the RNA, you have CUG repeats. And what happens when the number of repeats is too much, for example, more than 75, so what this CUG does, it binds to the MBNL, so which is a muscle blind protein, which is important for muscle movement. So what then happens that you're not able to move the muscles, you're not able to even control your movements, and when it goes on accumulating, for example, in case of Joshua, the number of repeats is 960, which is a terribly high number, and he cannot even smile because he cannot move the muscles. So you have a very life-threatening disease, and you are severely disabled. So, but one thing good is here that we know RNA binds to the MBNL. If we can then prevent RNA binding to the MBNL, what you can do, you can rescue the function of MBNL protein, and then it will be able to do what it wants to do. So that's where our interest was. So if we could find something which can say block this MBNL protein, so or which can actually block RNA, and then RNA doesn't bind to the MBNL protein, then you can get a small molecule targeting myotonic dystrophy. So for this purpose, what we did, we actually took a model system where we have a single CUG repeat, and we did some dynamics because we wanted to understand what kind of the motion is in the UU loop. And to our surprise, we were able to find that it can form one hydrogen, zero hydrogen bond, and two hydrogen bonds also. So a normal interaction pattern would be a one hydrogen bond, but it is also possible to actually have a significant proportion of the molecules with a zero and two hydrogen bonds. So that shows that it's not a simple sort of static molecule. It's quite dynamic molecule. And when you want to actually develop small molecules, you cannot ignore these conformations. So that's what we have. We have so many ensembles, and then if you want to do docking or you want to go for a virtual screening, you have to take the ensemble approach. You cannot just take one static structure. So I will show this movie where you can actually clearly see that, yes, you can see this movie. Sometime you have the one hydrogen bomb, then nothing, and then you have certain conformation where it comes closer and you have the two hydrogen bond. So I will just stop this movie and go to the next slide. Then we also took many repeats where we had 3 repeats, CUG repeats, and we were able to get the crystal structure. And in fact, in the crystal structure, it was possible to show that yes, these you have 0, 1 hydrogen bonds. So this crystal structure will be the sort of our starting point for if you want to do docking and all. But still, you have to consider other conformations of that one. So I will show slightly in terms of what are the strategies we use. So there are different strategies. For example, you can use microarray platform-based technology for targeting the RNA, where you can embed your ligand or your RNA on some solid surface, and then you can do a screening. And the informatics-based approach, which is a pretty interesting approach, where we use the information of RNA and small molecule libraries, and then fish out molecules which are selective for one particular sequence of the RNA. And I will also give a little bit of information on the fragment-based approaches. And then phenotypic assay, obviously, I will not talk about much, but for if you want to find, say, molecules which are active for bacteria, yes, this is a very good, interesting approach. So first, to show you about microarray platform-based. So for RNA, there are mostly 3 types, but you can develop your own ways. So one is like you have a aminoglycoside-based. What we do, you actually have glass slides where you deposit your ligand, and then you take different RNA molecules and do a screening to see which RNA binds to which particular ligand, and then you can get a sequence of that ligand and get to know the identity. Then you can have a peptide arrays or peptoids array. They also give you a lot of information. Then you can have an array of small molecules. I'll show you one example where what we have done, we have developed aminoglycoside platform for, say, look for small molecules which target this RNA. So you have this functionalized agar slice, you have this alkyne there, and to do a click reaction, we have ligand with a azide group, and we do the click reaction, and you get this ligand which is functionalized on the surface, and then we do a lot of screening with RNA molecules to find out what possible ligands are binding to the RNA molecules. This is a very good strategy. But there is an additional one where we have developed, say, these are the design ligand, and you can manipulate the backbone, you can manipulate how many units of the actual molecules are there. So this is a peptoid-based screening. So what we do is that we use peptoids. So peptoids are just simple peptoids, but what we have done, we have replaced the chiral group with a chiral methylene unit. So you get the peptoids. And then you can have any resin where you can put those peptides. So you link your molecule, then you pull them, and then you split them. So if you do this assembly, the split and pull synthesis, you can make a huge number of molecules. So the advantage for this particular technology is that you can assemble your ligands. So it means you can make demand ligands. So for example, if you want only 2 units of aminoglycosides, you can have that. You can actually modify your peptide backbone to increase the stability of your compound. And then you can put some of the cell penetrating groups. For example, you can put arginine tail on that, and you can increase the diversity, and you can also get as good as a natural product like ligand. So this gives you a lot of control. So only drawback is that the molecules are bigger, bulkier. So getting to a stage where you want to a drug to be approved by FDA is a little bit of an issue, because it then violates all the Lipinski rule. But it's a very good technology to find out the ligands, and then you can control. So obviously, when you want to target ribosomes and other those antibiotic, say, or antibacterial targets, this Lipinski rule is almost violated. So, but that gives you a control to develop compound. So you can have a one compound where you know what can be the sequence, or you can have you just do a blind things where you don't know about the sequence, but then you do a binding, and then you go back to the mass spec and you can figure out what's the molecule which was binding to RNA. So both are possible, and you can have a huge number of molecules. I'll show you later where we can actually extrapolate this technology. And just to show you some of the example where we have synthesized ligands based on the kanamycin, neamine. So these ligands then we tested for RNA, and to figure out what are the different RNAs they are binding. And as I mentioned earlier, that you can put a Arg peptide tag for cell penetrating properties. So that's what we have done in this slide, where we initially synthesize the say peptide ligand. You can see you have this typical solid phase peptide synthesis protocol, where you use the bromoacetic acid and the amine, go through various cycles, and when you build up your molecule, you can then put a Arg tag, and that will give you the cell penetrating peptide. So that's what we did. So in this publication, where we synthesize those ligands, these are huge ligands, yes, but they were very active in nanomolar concentration. So you can optimize what molecules you want, how many units you want. So it gives you a lot of control, and then you can increase the size of your library. So because you are going split and pool. So I will now come, and then I haven't given you all the example, but then you can look at some of the publications which I mentioned in those slides. So there are so many approaches which you can do. You can have a cyclized one. So we have done some of the cyclized one also, but I will come back to the informatics approach. So which is more close to what we do for the proteins, like finding small molecules. So because we have been dealing with the small molecules for a number of years now. We know them, we trust them better, and we know their pharmacodynamic and pharmacokinetic properties, and we can predict, we can actually manipulate small molecules biomedicinal chemistry, and that gives you a lot of control. So there's a lot of interest on that. So how our approach, how we approach the small molecules. So what we have done, we know what are the different RNA molecules. We have the information what are the ligands binding to them. So what we do is we have these databases. So I will show you one of the database. I will give you a demo which is online. So, but there is also this paper in chemical biology on this particular approach. What we do is that we take RNA small molecule database where we know the information about the ligands which are binding to the different RNA molecules. Then if we get a sequence of particular RNA which is responsible for that disease, so we get the information about the secondary structure of that RNA. So we can do thermodynamics and get information about what are the loops, what are the joints, what are the different sort of cavities when you go back to the 3D structure. And then we search in the library, so what that particular RNA junction, what molecules are binding. Then we have additional chemical library, and then we screen that to figure out what are the additional molecules which can bind to those particular RNA. And then we go about testing that, hoping that RNA inhibitor complex is formed, and this RNA is able to be being blocked by the small molecules, and it cannot form the protein. So that's where you can use this. So this is a very powerful approach, and there are a number of companies. I will mention later on what different companies are working on. For example, one of the cancer gene, MYC, is being targeted using this particular approach. So very powerful approach. So, but it requires you to have a database. It requires you to have a knowledge about the focused RNA library, and we have done a lot of work in that regard. So I will show you another example which we published way back in 2012 in JAX. So what we have done, we have taken that ligand database where we have a number of different loops, and then used the virtual screening to identify the molecule based on the shape similarity. And the shape similarity molecules which we used is pentamidine P and HOS. These are known to bind to the RNA, and we have used the information, 3D structure information, to figure out what are the different molecules which are close in shape and electrostatic to these molecules. And this was our protocol. So you have a database, you have a query molecule. So you look on 3D shape, you look on electrostatic, you look on pharmacophore. So you can pretty much develop your own descriptors. So then you do, say, shape-based or electrostatic-based or pharmacophore-based screening, and you get a hit molecule of 500. Then you can actually look at those molecules which are potentially in terms of, say, synthesis, in terms of IP, then generate a focus library that you can either purchase or synthesize, then go for biological assay, get the lead molecules, and then your typical medchem synthesis where you optimize the molecules. So I will show you one. This is one of the example. For our case, we wanted to actually see whether we can use this for finding small molecules which can disrupt the interaction of RNA and MBNL. As I told you in the earlier slides, that the CUG repeats are terrible. They bind to MBNL, and the muscle bind protein is not able to then do its proper function. So if we can have a small molecule which can bind to this particular RNA and disrupt this complex, you're able to say stop this RNA gain of function. So that way you can rescue the protein which can do the function. So we developed this assay, FRET assay, and where we have this terbium and XL65. There is a FRET going on between, because they are close together. So if you put a small molecule, it breaks the FRET, and you can then have a different signal. So by measuring the signal, you can actually you can say by FRET turn on and off, you can figure out how much efficacious your small molecule is. So we were able to do it for both pentamidine as well as host. As you can see, say, for example, pentamidine IC50 is 1000 micromolar, and one of our hit is 10 micromolar. So this is not, this is just by screening. We haven't yet optimized. So we further optimize this molecule. So that way, this is a very powerful approach. And we also did for the host, where if you look at the host, the IC50 is 400. but we were able to get a small molecule which is top 5057. So that was around 50 micromolar. So that is without any optimization. And we tested this molecule. In fact, we took top 5 hits to see what kind of pharmacophore is and what is the distance between those functional units. So that was around 15, 16 angstrom energy minimized structure of those compounds. And then we also tested whether these are specific for our CUG. And what we did, we synthesized a library of different RNA motifs where you can have CAG, CUG, so many different repeats, and the control also CTNT and human A site. And this molecule was binding specific to CUG. So we tested in the cell culture model, and you can see our molecule is able to rescue, say, when you have this, when there are no RNA CUG repeats, 65% of the exon 5 is included in that messenger RNA. So that's what actually our molecule does. So it almost functions as a control, which means you don't have any CUG repeats. And you can see also in that figure where we have the CUG repeat cells. So you can see these are forming a nice foci in that nucleus. And when you treat them with our molecule, it's pretty clearly when you have those CAG, they are disappearing. So this molecule is able to go inside the cell and bind to the CUG repeats. So that way it cannot bind to MBNA. So you don't see those bodies in the cells. So this was a very gratifying things for us to actually take an informatic-based approach where we use the information from the ligand to find out what additional molecules are active. So then I came back to India, and so the group there went back to going about dissecting the molecules, what different, how we can improve that efficacy. So I won't show you, but I will also show you one of the molecules for CAG, so which is important in Huntington disease. So we had that curie molecule DAPI, and so if you look at that, we were able to get a molecule D6 with the efficacy of 3 micromolar, which is quite interesting. So then we modified this molecule, but we tested this molecule in the Huntington disease fibroblast model, and it is pretty much able to rescue the binding of the CAG with the MBNA. So it means it is possible, yes, to target selectively. You just have to look the information of the ligand as well as the RNA. And I will also show you a little bit about the another approach, which is a fragment-based approach, and I think most of you might be familiar with the fragment-based approach. But just to show you a little bit of introduction. So say this slide is important in the sense that when you look at a molecule which is drug-like, so the molecular weight, if you take into account the Lipinski rule, is pretty much around 500. And if you have a candidate which is within this range, it's very difficult to optimize this because then the molecular weight will increase. But if you have a fragment where the molecular weight is around 200 to 300, it gives you a lot of scope. You can pretty much modify this fragment by so many iterative rounds and still be in that sweet spot of 500. So the fragment, they are not pretty potent, but those are small molecules which you can easily modify. And then you can combine those fragments with the linkers, screen them with NMR, ITC, biolayer, as well as there are so many MNT also nowadays available, and even mass spec. And then you have a different combination of those fragments. So you can pretty much cover a whole chemical space. And then you screen the compounds for what are the different different RNAs to which it binds. So, but you need a very good fragment database. So that will depend on what are the different targets of RNA and what are the small molecules. So we actually developed this database, smRNA database of small molecule modulators of RNA. I will actually just go and share with you, say, a little bit on this database. So just bear with me for a second. So I will actually show you what we can do there. So I hope you are able to see this is the Are you able to see something? Yes, so this is the smRNA database. And if you go into that search and give a textual search, so I will just look for the RNA. So like riboswitch, you go for riboswitch and give it a search. So you will see all the molecules. For example, there is adenine riboswitch, and then on your right, you can see all the molecules and with the KD and IC50. So this, you can do a text search, you can do a structure search, you can do advanced search. So you can pretty much know what are the different molecules which bind to that. And in that paper, so which we had published in nucleic acid research. So you can actually, there is a supplementary information about the fragment database which you can download, and then that you can use for your purpose there. So I will come back to this. So pretty much you can do all the calculations advanced, and our idea is to, so nowadays you have this SARS-CoV. So there are so many response elements you can target. So what are the different RNA motifs? You can find this information in this particular database. So now additionally, I actually, so I told you that small molecules are important, and the game is on the small molecule. But what is the size of the small molecule? I just want to share with you one particular slide, which I found very interesting. Is that there was a very nice book, Library of Babel, written by George Luis Borges. He was an Argentinian novelist, so he was blind. He actually imagined the universe to be a library, and all of the universe is filled with a hexagonal room which extend in every direction, and each room is filled with the books, and the books are randomly. So book can be any combination of letters. So he used 22 in the Spanish. So any combination of letters, and no two books are the same. And but this will actually, when you have any combination of letters, it will lead to a lot of garbage. And that's what he wanted to actually point out, that there will be a lot of garbage, but then you have an accidental combination of the letters which will give you some meaning. For example, you can find Imcheck in that. So if you go for a Library of Babel online, you can actually search that book, and you can figure out your name, everything in that. And whatever logical books he named, these are vindications. So the combination of the letters of these 23 letters will give you a huge combination of the books. The same is the universe, our chemical universe also. So I call it as a novum decimillion chemical universe. And if you look in that, the number of molecules, drug-like molecules, they are around 10 to the power of 60. The number of total molecules which can exist is huge. It's 10 to the power of 200. So, and the known molecules, which is the biggest database, is the zinc database. The known molecules is only 1 billion. So which is 10 to the power of 9. So it means we still have to go to 10 to the power of 60 to get a huge, to go to that drug space. And if you look at the biologicals, there is only 100 molecules. And if you look at the tools, there is 1,000. And just to give you an example, if you synthesize all the drug-like molecule and have a 10 mg of each, so that extends to 10,000 universe. And the number of stars in the solar system is 10 to the power of 50, around that. So it means our number of drug-like molecules are much more than the number of atoms in the solar system. So this is pretty much power of this chemical universe. We haven't even reached or scratched the surface, because one thing is there that you cannot synthesize those molecules. As I told you, that the biggest library is a billion molecule. So it's a very difficult to actually go and synthesize all these molecules, but there are ways. That's what I want to show you. What are the current technologies? So one of the ways is the DNA encoded libraries. So what you do in the DNA encoded libraries so for example, in this slide, you can see on the right, so we are doing encoding, and on the left is the chemistry going on. So you can build your molecule and you can build your DNA sequence on your right. Then you can actually say, for example, in next slide, in this particular slide, so what is happening, you are actually going through again the split and pool methodology, where you combine those libraries and split them again, then pool them again. So that way you generate a number of huge molecules. But interesting thing is that each molecule will have a specific DNA tag. So then when you take this molecule library and do a screening for, say, so many RNA molecules or protein molecules, and then you find a hit, then you can easily go for sequencing, and you will know about the identity of the molecules. So just to show an example, for example, we'll say if you do a ligation of the FMOC amino acids, so you can have 96 ligation, and by the step 4, you are able to reach up to 84 million molecules. So this is only by 4 steps. So imagine if you take it to a higher steps, you can pretty much go for so many molecules. And currently, the biggest library from DL is around 100 billion molecules. And good thing is these molecules will be in a small append of tube because they are in that bead. So what you have to do, you have to just take the append of tube, put all your RNAs, and then see which RNA binds to which molecule, then go and do the sequence. You will be able to figure out, yes, this is the particular molecule which is binding. So that's what we are trying here in the tech. Then you can have a encoded library, so you can have a peptide on one side, DNA on the other side also. So I will not talk much about that. But just to show you that if you have a DNA library, so what advantage it is there that you can actually go to the size of the cells. So you don't have to go for mass spec. You can do a flow cytometry and pretty much get to the sorting of the molecules, and sequencing you don't have to do. You can pretty much know by standard flow cytometry. Yes, this is the molecules which are able to bind, and then you do just a sequencing and figure out the identity of the molecule. So, and just to show you that this is another interesting slide. So where do we get our molecules? So if you look, 43% of the molecules we do by a random screen. But now this DEL, which is now 1% of the molecules are being figured out by using this DNA-encoded screening technology. So this is making a lot of inroads into the early drug discovery, where you get back to know about the leads. So just to show you also in terms of what additional RNAs we are targeting. So one of the particular interesting RNA is microRNA. So these are small 22 nucleotide RNA. So what we do, what are the challenges where we focus is in cancer, diabetes, and neurological diseases. So the challenge here is that as these are small nucleotide RNA, so you need to understand their 3D structure, what are the different hotspots, cavities. So we do a lot of molecular modeling to figure out the 3D structure. Then we confirm our 3D structure with NMR and MD. So you get to an understanding of what conformationally space they are occupying, how the bases are dynamic, what kind of loops are there, what kind of hydrogen bonding pattern is between the different bases. Then we do a docking to identify what are the druggable sites. So we use both flexible and ensemble-based docking, and then we develop small molecules. Just to show an example, for example, MA208 is important RNA involved in cancer. So once you get an information about the sequence, so you can do a secondary structure prediction, or you can pretty much do thermodynamics and get an idea about the secondary structure. Then you can actually do some base pairing modulation, what kind of different hydrogen bonds are formed, and get to the 3D structure. And once you have the 3D structure, so you refine, you get information about NMR and MD. And RNA is pretty much interesting. You can pretty much do a say very good NMR NMR spectra of RNA and get information about what are the different 3D motifs in that. And then you do MD to say refine and validate this model. Then further with computational medicine chemistry, you can understand what different binding events are taking place, whether it's electrostatic based or whether it's shape based. So you can pretty much use any small molecules, any peptide, any PNA molecules, then screening, synthesis, assay development. So this is what we are doing. So just to sort of summarize what our approach is for RNA. In fact, we go through a bottom-up approach. And if you remember Feynman's famous sentence is that there is a plenty of room at the bottom, and that's pretty much valid for RNA. So the idea is here that you get information about the genome sequence, where you have a sequence information about the RNA molecules which are causing the disease. Then you actually validate, say for example, you can validate by sRNA or by other technology, zinc finger or CRISPR-Cas, that yes, these are the RNA which are important for causing the disease. Then you go for thermodynamical properties to get an idea about the 2D structure of the RNA. So, and as I have shown you also, we have a lot of information now what different small molecule binds to different motifs. So you can get an information about these molecules which bind to different RNA motifs, and then you go and synthesize those molecules, and then pretty much you can do a say virtual screening, docking, and then get a ligand, and that ligand you can actually put on any peptide backbone or any solid surface to increase its efficacy. So that way you can actually design a modular ligand, you can design a high affinity ligand, you can design a specific ligand for a particular RNA, and also RNA are easy to manipulate in terms of synthesis, so you can have a library of RNA and you can let your ligand incubate with that library, so that the ligand chooses what it wants. So it goes to that particular RNA which it likes. So we just briefly few slides what we are also interested in. So we are interested in making nanoparticles from this RNA. So what we do here, we don't make nanoparticles from, say, for example, silver or other metal ions. We use this RNA templates to make nano carriers, and then we can decorate those nano carriers with drugs. So what we do is that we use the T7 polymerase to actually make a different RNA molecules. We purify them, and then you do a interstrand assembly by acid or magnesium chloride, so they form nanoparticles. You purify them, then you can have a different drugs, different moieties attached to that, and then you can deliver particularly at a particular place where you want. So this is this is where the RNA has an advantage, because it's a biological molecules. It can degrade fast also. So like other nanoparticles which are toxic. So we have used this database to find out what are the different motifs which can be used for assembly of those RNA nanoparticles. So for example, you can have CUG repeat, you can have tetra loop. So these are the particular things which we can use. So just in the end, I want to show you what is the future for RNA. So how RNA therapeutics is going to work out in the next few years. In fact, just to show you this slide. So I think future is pretty bright. So this is still a relatively new field, and but already a lot of inroad has been achieved in this. For example, Arrakis Therapeutics. So they started purely on focus of small molecules for RNA, and they are now able to raise 130 million. And Skyhawk Expansion Therapeutics. So this is a company of my postdoc boss in Scripps. So this is so it was incorporated in 2016. So when I came, I actually at that time in India, startup and other things were not there. So, but you can see. So I had a little bit of ideas that is the reason I wanted to work for their RNA, and we have collected a lot of information on what are the different kinds of the molecules. So I have shown you that database. In fact, it's one of the heavily used database. Every day I get a lot of requests for say the molecules or the RNA sequences which can be used. And just to show you, in April 2020, so Arrakis actually, they got an upfront payment of 190 million US dollars from Roche Diagnostics for RNA-based therapeutics. So future is bright. There's a lot of things to say. Still, we have long way to go. We have to understand a lot about RNA, how they bind, what is electrostatics, and the day we know about it, pretty much we can stop any protein. So the day you are able to say, yes, I have a small molecule which can block a particular messenger RNA, so that will be pretty beautiful, because then you don't have to worry about the protein. You just go and target that RNA molecule, and that pretty much will do the case. And this is happening. For example, I told you about Arrakis Therapeutics. They are focusing on cancer MYC gene, and they have a lot of data to show yes, they are able to selectively block the messenger RNA for that particular gene. So that is the power of developing, say, specific molecules for a particular RNA sequence. So in the end, I just want to show you one slide. It's a very interesting story which I always tell. Is that this is about Mullah Nasaruddin. He was a famous character in central India with his donkey, and so you can see from here he's looking up something, and then one gentleman on the top says, Mullah Nasruddin, what you are looking? He said, I'm looking for the key. So he also went down and started to look for the key, but he was not able to find out. Then he told Mullah Nasruddin what happened. I'm searching with you. There's nothing is happening. You haven't lost the key here. Tell me what is happening. He said, no, I haven't lost it here. Then the man got angry. He said, where you have lost? He said, I have lost near my house. Then he asked Nasruddin, why you are searching here? And Nasruddin answers, because the light is more here. So what we have been doing is we have been going and searching on the proteins because the light is more there, because you know the protein more, but we should go and search where actually the problem is. So looking at the RNA. So thanks very much for that. And I would like to thank my students who have been working, Saurabh, Isha, Ratan, Safi, Ankita, Vikrant, Vidya, Abhineet, Sonali, Sapna, and Sunil. So we have been working on different projects. So various collaborators, Matthew Disney, I actually initiated RNA when I was in Oxford, and we wrote a project where we got an interesting idea about targeting small molecules. Because in Oxford, I was actually interested in computational drug discovery, and that's how I got the idea about if we can target RNA. And Grant Churchill, of course, there is a lot of support from him, and Dr. Amit from IIT Indore. We have been collaborating on RNA, developing small molecule therapeutics for RNA. And of course, to our director, Dr. Sanjeev Khosla, for giving me the support, and IMTech, of course, where I'm currently based. Scripps, Wellcome Trust, Oxford, all the cash came from there. Without their support, we couldn't do it. DST, DBT, OSDD, so they are important funding bodies for us in India, and a lot of support from them to carry forward this research. And thank you very much for the talk. I'm ready for any questions. Thank you.
Thank you, sir. It was a really very wonderful talk. So I have with me now Lizo. He will be taking a few questions. Lizo, are you there?
Yes, sir, I'm there. Thank you very much, sir, for the most informative presentation of yours. Sir, we would like to ask you a few questions.
Sure.
And thank you, sir. So the first question is, what is the main, sir, kind of difference in CRISPR technology and RNA I technology, and how this will be useful for creating or discovering new drugs?
So see, you actually want to see, say one with the CRISPR is that you can have a very selective sort of blockage of that function. With siRNA, you can pretty much block other RNAs also. So, but CRISPR gives you the power to have a site specific. So if you want to understand, say, what is the role of that particular RNA in that disease, so you need to understand, you need to have something so that you can block it and you are not able to then see the disease condition. So this is the first. So, for example, I told you the bottom-up approach. So if I go to that slide, bottom-up approach, what we do is that you need that information. This particular RNA is the one which causes that disease. Then only there's a point in targeting. So that's where this CRISPR-Cas as well as this antisense therapeutics become important, because then you want to know, yes, you want to validate, yes, these are the RNA which are causing the disease. and they are responsible for that biological property. Then you go about discovering the small molecules. So what I would say is these are very important tools to get so that you know that target is validated. So target validation is pretty important. And for a protein, you can have different sort of techniques, but for RNA, these are two very important ones. So once you get the validated target, yes, then you look at say what information is available. That's where the RNA motif ligand database is important, because then you have to look at what are the different grooves or what are the different pockets in RNA which you can target, and whether there is any information already. So once you get an idea about, yes, these are the molecules, so either you can use that molecule as a starting point. So you can either do a docking to see, yes, whether it is docking at that particular place, because pockets, say one thing can be there that yes, obviously this RNA has a pocket, but then it has additional bases. So how those additional bases affect the structure of that particular RNA molecule, that is an important. That's where actually what we do is that we generate a 3D structure from say using the NMR data, and then we take those molecules and dock it, or take those say ensemble of RNA conformations and dock those molecules and get an idea, yes, this is that binding site which definitely is being occupied, and then we go about improving the ligand for that. I hope it answers your question.
Yes, sir. Thank you very much, sir. Sir, we will like to ask you a second question, and it is like since RNAs are being considered one of the major targets in the viral diseases. So
Yes.
currently, what kind of challenges you foresee, and what are those challenges that we have to overcome for these kind of RNA targets, so that we can design some efficient molecules to them?
Absolutely, I think I'm glad you asked this question. So one of the biggest challenge is to understand the electrostatics of the RNA. So RNA are not like a protein where you have a charged neutral thing. They are actually negatively charged molecules, and electrostatic, and their binding, the binding of a drug or a small molecule to RNA, in my view, is only governed by two things. One is shape and one is electrostatics. So these are the only two descriptors which we need. But then these are the only two descriptors for which developing force fields are pretty challenging. So you need to know the correct electrostatics of the molecule which is going to bind the RNA, and then you need to know the correct shape of that molecule. So that's why three-dimensional shapes where you use the Gaussian function becomes important. And electrostatics also, electrostatic is not a trivial thing. So there is, you can do Poisson-Boltzmann solutions, but these are approximate solutions. So once, I think one of the area where I would like to focus is that whether we can take a ligand and force it electrostatically to go to the RNA. If we can control that ligand moment, so that we know how the RNA is able to capture that ligand, then we will have a greater control over developing therapeutics for the RNA.
Thank you once again, sir, for your informative session, as well as for answering these kind of questions which the students have asked. Now I may request Dr. Mahanta to kindly give the concluding remarks for the session.
Thank you.
Yeah, thank you, Lizzo, for putting the questions. So as we come to an end to this really informative session, on behalf of the DDH 2020 training chairman, Dr. G.N. Sastry, and the entire team, I'd like to thank Dr. Raman Prakesh for taking this session and making it really a very important contribution to this entire training program. Thank you, sir.
Thank you very much.
And also all the viewers who are there in the YouTube as well as MS team during these sessions. And so we'll end up this session here, and we'll have our next vertical at 6 p.m. Thank you, everyone. Thank you.
Thank you very much.